So the next speaker is Victor Corces. Victor is a professor in the Department of Biology at Emory University. He's also part of the Cellular and Molecular Biology Research Program at Winship Cancer Institute. So Victor is well known for being interested in 3D chromatin, and he is studying the mechanism by which chromatin has a 3D organization and how this 3D organization is established and maintained. He published a number of landmark papers, one in particular looking at 3D chromatin structure in sperm cells, and he's used a lot of the method of ataxic. So today he's going to tell us about the mechanism of transgenerational inheritance of obesity epiphenotypes. Thank you, Victor.
Thank you, Isabel. Okay, I would like to sort of follow on the theme that Mike Skinner started to tell you to focus on the transmission of obesity. And we use the paradigm that Mike Skinner published a few years ago, and we use BPA. And so we expose pregnant females to BPA, specifically between days 7.5 and 13.5 of embryonic development. And as Mike also mentioned, for other exposures, the F1 generation seems to is normal, and we follow obesity, and so we follow the weight. This F1 generation has normal weight. The F2 generation is obese, and the F3 and F4 generations are also obese. And so these crosses are crosses between a mouse that came from an exposure in this female and another mouse that came from an exposure from this other female. Now we can also do outcrosses with control unexposed mice, and we observe the same phenotype. So if the F0 is the female that is exposed and the F1 is the progeny that was exposed in utero, if the F1 generation is not obese, if it's a male and we cross it with a unexposed female, control female, the next generation, the F2, now the progeny is obese, and if we continue these crosses, they continue to be obese. And so we can transmit the obesity phenotype either through the male germline or the female germline, and this will become important later on. Now, let me tell you a few things about the phenotype that I think are important in later on in interpreting the results that I'm going to show you. So the mice, when I say they are obese, they are actually larger in terms of size. The skeleton is bigger, the muscles are also bigger. They are obese because if we look at the visceral fat tissue, this is the fat tissue in a control mouse. This is in a mouse that comes from a BPA exposed individual. I'll refer to this mice always as BPA mice, but remember the exposure only took place in the F0 generation. When we look at this fat tissue, we find that the BPA mice they have more mesenchymal stem cells, which are the cells that will form the adipocytes, and they have the adipocytes are larger, as you can see in this figure here, and so they are storing more fat. So it's not only the body weight that is increased in BPA-treated animals, but also the percentage of fat tissue is increased with respect to controls. If we look at the weight during different generations, you can see the weight of the controls. The F1 is not fat, and then the weight increases and is maximum in the F4 generation, and then it starts decreasing such that the F7 generation weighs the same as the control. If we define an obese mouse as one that has a weight that is the mean plus a standard deviation with respect to the control, if that's the definition for obese, then some control mice are obese. The F1 generation is the same as the control, and then the percentage of obese mice defined as the mean plus one standard deviation in weight goes up to approximately 60% in the F2, and then 90% or 95% in the F4, and then it starts decreasing. So this means that the germline of the F4 generation is already, whatever epigenetic alteration happened to the germline is already going down. The F6 generation, more or less half of the mice are obese and half of the mice are not obese. And remember this because I'm going to come back to this issue. And then the F7 generation is not obese, it's the same as the control. That means that the germline of the F6 lost the epigenetic information. And you can see that if we look at the change in weight with age, when at week 3, the control and the obese and the BPA-treated mice are more or less the same, and the weight difference increases with age. And to make a long story short, the reason for that is that the progeny of BPA-treated mice eats more. And so in many of my slides, I'm going to compare control to, for example, BPA F4 generation, but I'm also going to compare mice from the 6th generation, from the F6 generation, the ones that are lean and the ones that are overweight, based on what I showed you here, okay? So if you compare the amount of food that the mice are eating, you can see that the control mice, which are the white, compared to the obese mice, which are the blue, the obese mice, the BPA mice, eat more during the light cycle, and not as different from the control during the dark cycle. So that means that the BPA mice, for whatever reason, which I'll try to explain later on, they are eating more during the day. So they spend the night eating and then they spend the day eating, and they don't, instead of burning stored fat, this is the respiration rate, instead of using the fat during the day, they use the carbohydrates that they are using in the diet. So the reason why they are fat is more of a behavioral issue than a metabolic issue. Okay, so one could argue that what we are seeing is a consequence of the environment of the mother. Although I told you that we can transmit it through the paternal germline, in order to discard this possibility, what we did was to take eggs from a F2 female whose grandmother was treated with BPA and a sperm from a control male, and we did in vitro fertilization, implanted the embryos in a pseudo-pregnant control female, so the environment of the mother now is not an issue. We analyzed the progeny. As you can see here, the progeny is still obese, the progeny from this cross compared to control. Okay, I should mention that the obesity is much more pronounced in males than females. In females, the differences are not very obvious. Okay, so that's the phenotype, and that I think is important to pay attention to the details of the phenotype because they are going to help us understand the molecular basis of the process. So let me remind you that we expose, this is mouse development. We expose the embryo between day 7.5, which is when the PGCs first differentiate. And then up to day 13.5. So this, we use this time because that's what Mike Skinner described originally, and is exactly the time when the germline is getting demethylated. So this diagram at the bottom is the level of methylation. You can see during this period of time, the DNA is becoming demethylated. If we expose the animals for only 1 day, at day 13.5, we see the same effects. We still see the obesity. If we expose them at day 16, which is somewhere here, after remethylation has already happened, then we don't see, they don't become obese. So we think that this window of development when the DNA is becoming demethylated is important. So what I'm going to tell you is that, so a number of years ago, we showed that the sperm maintains a lot of transcription factors, nucleosomes, in enhancer and promoter regions, and they maintain CTCF, which is a protein that is important to form these loops that you can see here in high-seq data, to form these loops that maintain enhancer-promoter interactions. And so what we think that is happening is that it's not just the methylation, but it's an interaction between methylation and transcription factor binding. So normally the DNA at this time is methylated, it becomes demethylated, and then is methylated again, and that's the end of it. Now, if at the time when the DNA is demethylated, you perturb the environment in the germ cells of the embryo such that you alter transcription, then some transcription factors that are going to normally are not present there or are not present at the same levels are going to be able to bind to the DNA at sites that are demethylated. And then when remethylation takes place, those transcription factors will protect the DNA from remethylation at that specific site where the transcription factor is binding. And we published a paper recently showing that this happens in the absence, normally, during normal germline development in the absence of any exposure. And the reason why this is important is because if the environment alters the binding of transcription factors to the genome, and then those transcription factors protect from demethylation, that demethylated DNA now will be able to interact with the same or other transcription factors later in development. So what is important about this is that, and we showed this in a paper that we published last year, that binding sites for transcription factors that are present here, you can detect them through a tag-seq. Binding sites for transcription factors in the sperm and in the oocyte, a subset of these binding sites for transcription factors are maintained in the embryo through all embryonic development, so that the germ cells can pass on to the embryo information through the presence of transcription factors bound to DNA. What is important is that these transcription factors are bound to enhancers, and they are mediating enhancer-promoter interactions and forming loops in the chromatin. And this is a meta-analysis of high-C in sperm and throughout embryonic development. And you can see that these dots represent a collection of all the loops mediated by CTCF. You can see that these loops present in a sperm but not present in the oocyte can be transmitted to the embryo, okay? So what we are trying to explain essentially by looking at the transgenerational inheritance of a phenotype is how information that determines the expression of a gene in the germline can be passed on to the next generation. And so what I'm arguing, and I'm going to argue, is that it's in the form of transcription factors bound to DNA and interactions between those transcription factors and promoters. So we can use a tag-seq to look at changes during during germline, in changes in the germline during different generations of this exposure that I mentioned before. So for example, these are all the ataxic sites. So when we look at mice, the germline of mice exposed to BPA in different generations, we see very few losses of ataxic signal. And in all the experiments that I'm going to tell you, we do ataxic and we select specifically for fragments that are between 50 base pairs inserted in the reads, 50 base pairs and 118. And so these are regions of the genome that are protected from TN5 insertion by the presence of a transcription factor. So all the ones that are gaining BPA, so these are all the sites in control, and they are gaining BPA in the F1. So this represents what happens in different generations to the ones that are gaining F1. So you can see that some of them stay and some of them are lost. And out of all these transcription factors, we can look in the ataxic signal and look for motifs. Many of them correspond to CTCF. So all these are CTCF sites that are normally not present in the germline in the sperm and now are present in the F1 after BPA treatment. And some of them are lost and some of them are gained. Now, if we look at what happens, the consequence of this transcription of this new CTCF sites, you can see, so these are the CTCF sites that are increasing BPA, and you can see that they form new loops. You can see it here in high-C data. So this is control, this is BPA. This site here was not interacting with this site over here in control, but is now interacting in BPA, and that corresponds to this CTCF site and these CTCF sites that are increasing in BPA with respect to control. And when we look at all the CTCF sites, and this is a meta-analysis of all the interactions mediated by these new CTCF sites, you can see that there is a dramatic increase in interaction frequency, meaning the stability of the loops. And this is important because CTCF targets enhancers to their cognate promoters, and also they form loops and they interfere with the ability of regulatory sequences inside and outside of the loop to interact with each other. So you can see that in the F1 sperm, there's a dramatic change caused by BPA exposure. Now, if we now, you remember that the weight increased up to F4 and then it started decreasing. So now if we require that these sites are not present in control and they are present in the F1 to F4 generation, we find 69 sites. Okay, and they are mostly present in distal intergenic regions and introns. So they correspond to enhancers. And when we look at the motifs that are present at those 69 sites, you can see CTCF is the one that is present at most sites. So most of these new sites have CTCF. And the ones that don't have CTCF have zinc finger 143, which is a protein that colocalizes with CTCF, FOX transcription factors, which are pioneer transcription factors that are required for recruiting nuclear hormone receptors to chromatin, and then a lot of nuclear hormone receptors, estrogen, androgen, estrogen receptor, related alpha and beta, PPAR gamma. So these are the 69 sites that are present consistently in the F1 through F4 generations. And so this is the actual data. You can see this new CTCF site that was not present in control and is now present in all the different replicates of, I only have up to F3 here. Now, I told you before that what we think is happening is that these transcription factors are binding to the germline, and they, at a time when the DNA is demethylated, and that they protect from demethylation. So we've looked at changes in DNA methylation through in the progeny of in the germline of different generations, and we have identified a lot of changes in DNA methylation. So these are hypermethylated regions. These are hypomethylated regions. You can see them here too. Most of them are located in distal intergenic or in introns. Presumably, they should be enhancers. However, most of them don't overlap with ataxic sites. So this is something that is sort of puzzling, but it would suggest that these DMRs are not affecting the binding of proteins to DNA. However, when we look at these 69 sites that I told you are conserved, and we look, this is the increase in ataxic signal from F1 to F4, and this is the change in DNA methylation. And this here tells you the number, the size of the circles tells you the number of CPGs, and CTCF sites are in orange. And so if you look at the hypomethylated regions, so in all the regions that are below 0, you can see that it's mostly CTCF sites that are affected, okay? So this is an example, for example. Okay, so let me tell you, out of these 69 sites, 33 decrease in DNA methylation, 22 actually increase in DNA methylation. So those 22 are these guys here, and 10 are not changed. So this is one case, just to give you an example. It's a CTCF site. It's present, the CTCF site is present in BPA sperm, but not in control sperm. And this region is hypomethylated. So this is BS-seq, and we have a very high resolution in the data, and you can see it's hypomethylated with respect to this. Now, if we now add another requirement for this site, so we have 69 sites. Now we require those 69 sites to go away in the F6 generation, in the germline of the X6 generation, because the F7 generation is not fat anymore. And so we end up with this, I think it's 12 sites. And one of these 12 sites is FTO. And so I'm going to use this. This is the FTO gene as an example to tell you what we see. So this is the FTO gene. As you know, FTO is the gene that has been best associated with obesity in humans, because SNPs in the FTO introns, which are full of transcription factor, binding sites, and enhancers, associated with obesity in humans. So we originally found, control disappears. And there are other sites here that correspond to FOXA1, estrogen receptor, androgen receptor, PPAR gamma. Are you guys still with me? I just got a message that my internet connection was not working.
Yeah, you're back. You're back. You went out for a few seconds. Yes.
Okay. Okay. So there's a whole bunch of different transcription factors in this region, and you know, there in general there's an increase in the BPA with respect to the control and with respect to the F6. Now we can also look at this embryo transfer experiment, and we see the same increase in this ataxic signal. Now, what is important, and I want to emphasize this, is so we've looked at the DNA methylation here. So we can see by BS-seq, we can see this hypomethylation. But if we look by sulfate sequencing, CTCF, the motif, has a C at position, a CPG at position 2 of the C. And this region is methylated in control, becomes completely demethylated. This is specific C in F3, and then when we look in F6, it becomes methylated again. Now, we think that the gene responsible for this is FTO because all these other genes are not altered in ovaries, in oocytes. So I told you that we could transmit this to the female germline, And so whatever changes are happening have to happen in the oocytes also. And the only changes that we see that are the same in sperm and oocytes are in the FTO gene. So based on this, we narrow down all this collection, this 12 genes, to just FTO. And so when we analyze ataxic high cyst, and we look at BPA and control, these are interactions that are present in BPA but not in control between this CTCF site that we find in the intron of the gene and the promoter of FTO, the promoter of this other gene RP-GRIP1L that is involved in obesity, IRX3, which is also involved in obesity, IRX5, and other genes in this cluster here that are also involved in obesity. So the presence of the CTCF site increases interactions between these enhancers in the intron and other genes in the vicinity. And so this is important because I told you that this configuration, not only the transcription factor, but the interactions can be transmitted to the next generation. Now, what complicates everything, I think I should be done, no? What complicates everything is that FTO is that an M6A RNA demethylase. And so we see a huge decrease in M6A methylation in sperm RNAs. And most of these decreases are in intergenic and promoter regions. I'm going to go very fast. And importantly, we found a dramatic decrease in an enhancer RNA, decrease in M6A methylation, not in the level, but in M6A methylation. And this RNA is present. Here is FTO. This is the enhancer RNA. And this RNA is associated with chromatin. So if you purify chromatin-associated RNAs, this enhancer RNA is tightly bound to chromatin, and it causes an increase in accessibility in this region, as was described in a paper in Science a few weeks ago by Chuan He's lab, showing that if there's a decrease in M6 methylation in enhancer RNAs, there's an increase in accessibility. So there's an increase in accessibility, and the proteins that bind to these accessible sites are CTCF and androgen receptor. So I'm going to finish here, just leave you with this message. So this is the FTO region, IRX3, IRX5. We think that what happens is that FOXA1 binds in this region, and BPA binds to the estrogen receptor, and there was a bind site in this region, and it binds during germline development, and then recruits CTCF, and that increases interactions in the germline with the adjacent genes. Now, FTO encodes this demethylase, and that demethylates the enhancer RNA, and then the enhancer, this region becomes more accessible, and it can recruit all the transcription factors, probably the androgen receptor based on the ataxic information, and that now increases interactions with the neighboring genes. Now, there are several interpretations of this. One is that whatever happened in the gametes, everything that I've told you, is transmitted to the zygote, is transmitted early during early development, the bound transcription factors, and those transcription factors bound are transmitted to the precursors of the germline during gastrulation. And then after this, one possibility is that that information is transmitted to all the adults in the somatic tissues of the adults, or not, or that alters transcription early in development of all the genes, and that is what is transmitted through embryonic development. Now, in addition to the transcription factors, then the change in M6A RNA methylation is going to affect other processes in the embryo. So there is this dual thing. now I have to, I won't be able to show you all this, except that just to come back to the end, that in what is happening in the fat tissue, we think, is an overproduction of leptin that is causing this behavioral changes in the eating behavior in the mouse. So most of this work was done by Yoonhee Yang in the lab, and other people that contribute to this are Bree Bixler and Daniel Ruiz and Xiaoling Wang. And I'm sorry I went over, and thank you.